I'm going to first introduce Yoni, because this is the International Visiting Professorship and Lecture for Thomas Jefferson University for Yoni. It honors him for all his years of service, and we're delighted that he gives this lectureship and chooses someone for it each time with PXE in mind many times, and much of the work that he has done that's really contributed to better lives for patients. So Yoni, turning it over to you.
Yeah, thank you. Good morning, good afternoon, good evening, wherever you are. I'm extremely pleased to introduce Professor András Váradi as the keynote speaker of our PXE International meeting. And let me see if I get the desktop somehow. Well, Amir, can you get that out? Amir is my able assistant. With the first slide, yes. Okay, so just a little bit of background. So some of you may know I have been 34 years as a chair of the Department of Dermatology and Cutaneous Biology here at Jefferson. And when I had my 30th anniversary a couple of years ago, the dean suggested to paint my portrait. Now, I don't like portraits, so I told him that I'm not dead yet. But how about having a nice professorship, visiting professorship and lecture, so I can invite my good friends and co-workers to Jefferson. And András Váradi is the fourth Yoni Judo, MD, PhD, International Visiting Professorship and Lecture. And unfortunately, András, you cannot be here and have one of those wonderful dinners because of the situation, but we'll make it up another time, either next year in Budapest or again year after here in Philadelphia. So Professor Váradi hails from Budapest, Hungary. He earned his PhD in 1975. And right after that, he became affiliated with the Institute of Enzymology of the Hungarian Academy of Sciences, where he has progressed first from postdoctoral fellow, then to full professor, head of a laboratory, and is currently emeritus professor. During his time at Institute of Enzymology, he also took time off to be a visiting scientist at Cornell University in Ithaca, 1983 to 85. He was visiting scientist at Yale University from 1988 to 1991. And he was a Fulbright visiting professor here in our department at Jefferson in 2008. András is extremely well known for his pioneering work of APC transporters, and he has made tremendous contributions to our understanding of the disease that we are discussing today, namely pseudoxanthoma elasticum, or PXE for short. So when I asked András to be the recipient of this award, we were very pleased that he accepted our offer. And we are very pleased to hear his presentation. Now, András is a good friend, and he has a good sense of humor. So what you see now in the screen, that's the Jefferson official announcement. See how everything is proper. Title is always look on the bright side of science. But András said, well, let me make my own announcement. And here is the kind of announcement of this lectureship that András made himself and sent to us. So as you can see, always look on the bright side of science. Professor András Váradi, please.
Thank you very much, Yoni. I am, how should I say, really moved. I try to share my screen first, but so I really feel honored and delighted to do this lecture. And And Yoni, would you do me a favor? May I ask you to do me a favor? Would you be so kind and briefly write this down and send to my chairman? I think it would help me a lot.
I'm happy to do it, but more importantly, I'll write it to your wife, Julia.
Good. It's even, yeah, you are right. It's more important. Okay, so you showed almost fully my lecture because that was the beginning and the end. So I start, and indeed I picked up a little bit funny title for my talk. Always look on the bright side of science. I do know that science has dark side and bright side, and you cannot be always on the bright side, but fun comes when you are. And actually, I cannot feel better joy than when you discover or you have told about a great discovery and you are surprised and you are happy. And I like to be surprised and happy. And it comes very often from science. Usually a keynote lecture goes like the lecturer gives an overview or gives a great review of the field. I apologize, I will not do that. I will not do that because I cannot do better than these two papers published about a year ago. They are great, they are critical reviews, deep, actually I would say entertaining or exciting to read. So I just ask you to read this. I cannot compete with those, even if I try to do my best. So rather, I invite you for a little journey. First, we will leave the field of PXE and transporters and our field. And I want to show you maybe two different examples of non-typical way of thinking in science on ideas which did not come first, but became successful and interesting. So let's go back to the Second World War. During the Second World War, Navy experienced a big problem. Bombers coming back from the war, and they were damaged. There were bullet holes all over. You can see where the red dots are. And they decided that they should do something. They invited a few smart people, engineers and scientists, and of course soldiers, to discuss how to protect better the bombers. So they made the plot, they showed the clusters of bullet holes, and of course everyone at the beginning told that, all right, here are many, here are more, even more here. We should add protection to these areas. But fortunately, there was a very smart guy on board called Abraham Ward. Abraham Ward was a mathematician from Columbia, and he said something that, wait a minute, we are on the wrong track. We are looking aircrafts, bombers, which survived the mission. And if they survived, they were not damaged fatally. So we have to concentrate on the areas, what I show you now with blue circles. Those are the areas which are sensitive. So we have to add more protection. Why? Because those are the areas, if hit, would cause the plane to crash and never come back. And this led to a phenomenon in statistical analysis called survivorship bias. And it happens sometimes, but why I show you this, and we'll show you another example, because I want to demonstrate that sometimes in science, what is trivial or looks trivial at the first minute, not necessarily true that that will give you the answer to the question. So we go closer to our field. Here is a genetic disease. I'm sure Yoni knows a lot about that. I don't. I want to use it just as an example. This is Dyskeratosis congenita, how do you pronounce it? Anyhow, there is a gene called dyskerin, and if it's mutated, the disease develops. So let's suppose that you have your students, you show them this cartoon or this slide and ask that, what do you think? Where are the essential domains? And of course, most people or most students would show that, of course, it's here and here, because these are the areas of the protein collected many mutations identified in patients. And if the mutation affects this area, there is a problem, there is the disease. Unless you know what Abraham Wald told, and I showed you before. So this might be wrong. Actually, the essential areas of the protein are here. Why? Yes, this is another example of survivorship bias. I use it that sometimes you have to think on the less trivial way as everybody would. Because if the mutations affect this or this or this, it will be fatal. You never will see those mutations in patients. So I just wanted to use these examples out of the PXE field that science is not unidirectional. Progressive science is not always very simple. What I want to go ahead on is a time chart of the history of PXE science. Here, the blue lines is the time. The blue lines is straight, always goes ahead from left to right. But the progress in research is not that straightforward. So I want to start in 2000. Actually, that's the year when I started to be interested in PXE. In 2003, different laboratories in Honolulu, in Philadelphia, and in Amsterdam published that they found or identified the disease gene of PXE, and the gene is ABCC6, encodes a transporter, an unknown, that was a yet unknown transporter. Nowadays, we have a clear or almost clear imagination how it looked like. It's in the plasma membrane, and it's a huge protein membrane embedded. But definitely this discovery was a surprise, an unexpected surprise. So I remember that Yoni several years ago told me that immediately when the paper came out, he walked over to the Fox Chase Cancer Center to see Gary Crew. Gary was a tumor biologist. Unfortunately, he died a few years ago because of an accident. And Gary was interested very much in ABC transporters, did a great job because ABC transporters, some of the members of this transporter family is associated to tumor multi-drug resistance. So when Yoni presented this finding, this great finding to Gary, Gary thought, very unlikely. And of course, Gary was not right. This is the disease gene. But that was I would say, a spontaneous and understandable reaction, because what the hell a transporter gene would do with extracellular matrix proteins? Everybody expected that this is a skin disease, maybe a structural protein we know in the extracellular matrix is mutated, or an enzyme that decorates or works on those proteins. So that was one of the first surprise. It happens. And immediately people started to look for the protein, or actually at the beginning for the mRNA in the tissues what are affected. There were fibroblasts, for instance. So it was looking where the symptoms are. That was logical, but they did not find, or we did not find the message there, because ABCC6 was not expressed in the dermal fibroblasts, although again, it was an extremely logical step ahead. 2 years later, when very good specific monoclonal antibodies has been generated in Amsterdam by George Schaefer and Rick Shepper, they clearly showed that the protein is expressed in the liver mostly, and to some extent in the kidney or lower level. So one question has been answered, where is the protein? But a more important question was arise. What the hell is looking a protein in the liver while the symptoms are all over in the body, in the eye, in the skin, in the arteries, but definitely not in the liver? And people started to speculate to find out the bridge between a liver transporter and a systemic disease. And actually, it became possible when knockout mice model became available. First of all, thanking to those models, we got a very nice, clear biomarker classification, but more importantly, going to the mechanism, Yoni's laboratory published 2 great papers showing or proving that PXE is a metabolic disease. Metabolic disease. And they demonstrated that should be a metabolite most probably coming from the liver. It's present in the circulation. It can be transferred from one animal to another, from a wild type to a KO. It's active, it's in the circulation. So I think that by probably in 2010, it was clear and nicely proven that PXE is a metabolic disease. Now it started a big race or hunt for this mysterious factor X. What is the metabolite which can prevent ectopic calcification or which is missing in PXE? Actually, I hope I remember well that at one point, Olivier told me that they do metabolome analysis with a great collaborator in Honolulu. Once I talked to Yoni was in Germany visiting someone maybe in Freiburg, if I remember well, collaborating with another great lab doing whole metabolome analysis. We tried to do something similar. We expressed ABCC6 in insect cells, generated inside-out vesicles. This would be a transport-based approach. And we thought that if there is a mixture, a biological sample containing lots of compounds, but includes this mysterious factor X, our transporter would transport it inside the inside-out vesicles. It is sealed, and we can isolate the vesicles and analyze those with the aid of good chemists, HPLC, mass spectrometry, and so on. Of course, we needed to add ATP because the transporter is an ATP-driven machinery. And we ended up to nowhere. We did lots of work, always ATP came back. Of course, ATP was sticking to the plasma membrane to the membrane of the vesicles, so it was a failure. Fortunately, Kuhn had a much better designed metabolomic study, which yielded to a great result, ended up with a great result. He used cells and analyzed the supernatant of cells, expressing or non-expressing ABCC6. And this is the story what everybody knows. Somehow ATP is delivered by the aid of ABCC6 from hepatocytes to the circulation of the liver. It's converted to pyrophosphate, and pyrophosphate itself is the factor X what can control ectopic mineralization. So here is a cartoon, and on the left, we are in the liver now. On the left, there is ABCC6, facilitates trinucleotide ATP transport from hepatocytes to the circulation. On the same membrane compartment, there is an ectonuclease called AMPP1 that converts ATP to pyrophosphate, pyrophosphate gets into the circulation and prevents hydroxyapatite formation, prevents or controls calcification. And here are the two genetic diseases due to the lack of ABCC6 or EMPP1. It's very straightforward, but that's due to the great discovery of Kuhn's metabolomic analysis. Okay, if we go back to the time chart, and we are now in 2013, 14, 15, something like that, you can ask, what is next? Of course, next is intervention. There were always great ideas how to do something with PXE, but now in those years, due to the discovery of the ATP pyrophosphate pathway, we know that we have a metabolite in our hand, which is simple, endogenous. We know how it works, so it's an ideal candidate for an intervention activity, or to develop a drug based on this discovery. Unfortunately, there was a general view immediately when pyrophosphate came into the focus of PXE research and the disease. And it said that pyrophosphate is a great discovery. I fully agree, but it will never be considered as an intervention. Why is that? It is because the intervention in this case should be oral, since we are dealing with a lifelong disease, so the treatment should be lifelong, and that can be realized only if you can deliver the drug, in this case pyrophosphate, orally. And there is a big however. There were papers even from the 60s. Here I just cite one from 1969, or more than 50 years ago, telling that pyro and polyphosphates are not active when given orally. And this is a couple of animal experiments what showed that. And almost 50 years later, there is another paper, a review paper actually from 2016, saying that pyrophosphate has to be injected as it is effective orally because of hydrolytic destruction within the gut. So we have a problem. These statements are very strong. There are animal experiments supporting that it is not active when it's given orally, and we know the reason why not. Here is a very strong argument that it will be hydrolyzed in the gut immediately. There are enzymes splitting pyrophosphate to inorganic phosphate fast. So it looked like hopeless. But somehow I had a feeling that let's give a chance to pyrophosphate. Give pyrophosphate to our calcifying animals on a way that we give them into the drinking water. And let's see what happens. Maybe it helps. And in the previous meetings, maybe more than once, I showed you that it works. The answer is yes, orally given pyrophosphate inhibits calcification, prevents mineralization. I don't want to show you all the experimental results. Actually, there will be another lecture today by Viola. She will show you the Vibrissae calcification. I just picked up one example. Those are the arteries. This is a whole arterial system of mice. We will concentrate on the leg, the iliaca, and I show you enlarged fragments, enlarged pieces of those arteries. Alisan staining shows heavy calcification if we do nothing. If we put the mice on pyrophosphate in the drinking water, you can see much less calcifying areas. Not zero, but a huge prevention. And the calcification, if we do not treat, clearly visible in the media layer of the artery, while also much less in the pyrophosphate. So it shows that what we see here is the calcification, which is characteristic as an ectopic arterial calcification. So it's an arteriosclerosis. I learned it not very long ago. You can quantify and it is clear. So we have it. Of course, if it's active, if it prevents calcification in mice, it is absorbed. And it is absorbed in human as well. We invested a lot of time and effort to do pharmacokinetics, basic pharmacokinetics, looking for the optimal salt form, optimal dose, optimal delivery method, formulation. I call your attention again to Viola Stoke. Viola will explain you all the details where we are now in human and how the mice absorption study support what's going on in human. So I think that we are in a relatively good shape in that. Hopefully a clinical trial is ahead in the near future. And if we are talking about clinical trials and interventions, I put together on the same chart as I showed you before, the different efforts. I put here only where we can, there are in vivo experiments, successful experiment, proving that the idea may work. So Kuhn comes later when you do something influencing, for instance, ONC expression, and there will be a mouse model and so on and so on. That's extremely encouraging, but I left it out because of the lack of animal model at the moment. But as you can see, most of the efforts somehow go to get enough pyrophosphate in circulation to be able to halt crystallization of hydroxyapatite, or other words, to prevent gasification. It can be directly adding or supplementation of PPI, what I prefer, and there are a European consortium thinking about that and pushing ahead a clinical trial in this field. Also in the Netherlands, pyrophosphate analogs has been, or one analog has been used before, and that will be continued. On both ideas, there will be talks today. There are other ideas to make the protein as a better transporter, or to have the protein to be expressed in the liver. And there will be a talk about tissue non-specific alkaline phosphatase inhibitors. Also, this is a more or less indirect way. If you can stop this enzyme, it won't be able to hydrolyze that effectively pyrophosphate, and pyrophosphate concentration goes up. So the magnesium is the only exception. All the other ideas today, including ANC, is somehow focuses on to get enough pyrophosphate in the circulation. So this is where the science finally not ended, but where the science is now presently stay. But we have new problems. And of course, that means that science won't stop. What I did, that I just went through the abstracts of the present meeting. What we received the abstracts yesterday. So here is the title of an abstract. Actually, that's a collaboration of my group together with the Wilco's group in Utrecht, and the title is Decreased Plasma Pyrophosphate is not associated with disease severity in PXE. So it's to me, it's a little bit surprising. It's very interesting. I don't think we understand that. What we know, that PXE is much more complex than simply one metabolite calcification and goodbye, science is done. But I kept reading. Here is another title. We will hear both today, which says that it correlates with the cardiovascular phenotype. So it's not associated or correlated. We will see. We will listen to these two talks and hopefully we will know. But then I kept reading the text of these two contributions. This says in PXE, that's just how should I say, not the major result, but it's interesting. In PXE, pyrophosphate positively, plasma pyrophosphate positively correlated with age. Interesting. Pyrophosphate were not influenced by age. Even more interesting. We will see what's going on. Now here is one unpublished result from my lab, which does not mean that this is the case, but we have a nice piece of data of healthy volunteers where the association between age and plasma pyrophosphate level is very strong. There is a very strong p-value, which not necessarily means that we did the experiment on the right way. Maybe the sampling is not optimal. So we don't know. There are lots of things what we don't know, and that makes life exciting. In addition to that, we have new players. I already mentioned UNC, but there are hematopoietic cells, blood cells. Olivier showed that they also contribute to some extent to what ABCC6 does and what happens when ectopic calcification starts to develop. And also, I found it very exciting. This is not new, it's always around, comes up and down this question. What is our job? I mean, as a scientist in the lab, at the bench or in the animal facility, so doing science. Who should translate the new results to intervention? That's always to what extent we have to go ahead or can go ahead. And I think that, I'm pretty sure that Kuhn, who ended up with this very important discovery of the role of UNC, will, or already started to see how can we or he influence the transcription of UNC. If you can go up with the UNC, you can get more pyrophosphate and you can replace the activity of ABCC6. Great idea. Or yesterday there was a kind of discussion whether in our work when we figured out that pyrophosphate can prevent calcification, the trauma-induced, accident-induced calcification, even when added after the accident, how can it be developed to an intervention? And I think we should try to do that as far as we can, as far the animal models allow further science and so on. So that's also exciting. At least it keeps me excited. So if we want to see the trajectory of PXE research, I like to bike, so I take an example from biking. Sometimes it goes uphill. It's enjoyable, but it takes longer time, more effort. Downhill, also enjoyable, much easier, much faster, but there is no downhill without uphill. And sometimes we go much faster than bicycle. Sometimes we feel that we are on an aeroplane. But I'm afraid that even the aeroplane make loops, not fly directly. Or we have a strong sports car. It's powerful, but we are on a snowy highway, so we cannot drive as fast as we want. But that makes it interesting, at least. So I found a very nice or entertaining movie short, what I want to share with you. This is an extremely complicated interesting traffic situation in England. It is real. This is a roundabout, and let's suppose that the yellow car is PXE research. It is in a chaos. It's so difficult to find how to get the right way, what to do. But our little yellow car finds a way and goes out and solves the problem. And personally, I think that we are not in such a chaotic situation, but complicated enough. Well, that's fun. So this is one of my latest slides. I want to show that somebody says that question everything. And yes, sometimes you have to question everything. Maybe not always and maybe not everything, but that's the art of science, when and what. So the people on the right, my group and our collaborators working hard on the pyrophosphate business or pyrophosphate project in the last, let's see, 5 years. Here are the collaborators. Already we started to work together in human absorption studies, including American and European friends. Pete, Bors, Balázs Sarkadi, Gergely Szakács, and Vin Arias were always around us, giving good advice, sometimes harsh critic, but it was always helpful and cannot be thankful enough. And we got support from Hungarian and American foundations for our research. The next slide is somehow personal. I told you that about 20 years ago, I entered to this field. And I had, of course, many failures, some results, lots of fun. But one of the fun or one of the enjoying situation came that I met great people. I met great scientists, enthusiastic persons. It was almost so much fun to interact with those, to exchange ideas, to debate things, even not only on science, but on political life, everything about world and universe, and so on. Sometimes we were competitors, sometimes we were collaborators, but always the friendship, the good connection, the great atmosphere was around, and I really feel gifted to meet them. So thank you. And then we enter to my last slide. This is nothing more than an advice. Always look on the right side of science. Thank you very much.
Thank you very much, András. Terrific, insightful summary of the PXE field. Thought-provoking, and I'm sure there are many questions which I believe they will be answered by the upcoming presentations. András, it's been my personal honor to have you as a fourth Jouni Uutto MD PhD International Professorship and Lecturer Award recipient. And we have prepared a little plaque type thing, not the kind of regular plaque. This is this kind of little thing, and it has an engraving which says Jouni Uutto MD International Visiting Professorship and Lecturer 2020 recipient András Váradi PhD. Now, this it looks very simple, but the point is that there is a globe which goes on top of it. And this globe keeps turning as long as there is light. So we always will be looking for light and the world will be turning. So thank you very much, András. It's been a great honor. Thank you.
Thank you very much. Thank you.
And Sharon, you can pick it up from here.
Right. Thank you very much. I want to also thank András for your work all these years. I recall going to your lab all those years ago to sit with you to say, can we do something here, given your background? And Jouni, of course, always, every year, very happy that we can be part of the celebration of all the years that you have worked on PXE and other diseases that are important to people. So thanks very much.
Thank you. Thank you for the opportunity. And thanks for the support, both emotional and material.
Right.
Thank you.
Wonderful. Good.